This recording is going to look at two passive transport processes. We're going to discuss simple diffusion as well as facilitated diffusion. So this is where we are in the lecture, but we're only going to do a couple in this particular recording. So we'll start with simple diffusion, but actually just remind you what passive transport is. So passive transport, there's no expenditure of energy. Substances are going to go from a high concentration to low until it reaches equilibrium. So it's going to be equally distributed across. So we're going to start with simple diffusion. So this is a movement of a substance from a region where it's high concentration towards low concentration. So it's going down a concentration gradient until equilibrium is reached. So let's just look at the scenario where they're putting dye into a beaker of water. So you start here where you drop the dye molecules. It's very high concentrated right here, but it will diffuse, and as it diffuses out, when it reaches equilibrium, it's going to be evenly distributed across that beaker. Now obviously if I put enough of that dye in there, the color of the water is going to be the color of the dye. The reason why these molecules are moving is they're moving. Molecules are always moving. There's constantly in motion, and they move by random molecular action, or I should say, they call it Brownian movement. So things are moving, and they're just going to keep moving until they kind of are evenly distributed. Well, what about the simple diffusion across cell membranes? So remember, cell membranes are selectively permeable. Now, an important thing I want you to realize is that cell membranes, or I should say only living membranes, like the cell membranes we have, can change the permeability or select for or against a particular substance. You don't see that with, say, dialysis tubing. You can't selectively change for it. With cell membranes, living membranes, you can. So let's just kind of go back. And so I want to mention about the transport of by simple diffusion across cell membranes. So this picture is illustrating where you start with a high concentration of some molecules, and they're going to start moving across that membrane from high to low until you see it's reached equilibrium. So the concentration's the same on either side of the membrane. Well, what type of things can move by simple diffusion? So these things, in order to move just directly across that membrane, they must be lipid soluble. So they're lipid soluble molecules, such as, say, steroid hormones. Testosterone is an example of that. Gases too, like oxygen and carbon dioxide. So those substances move across the membrane from high to low until it reaches equilibrium. Let's give you an example, real life. You breathe. The oxygen that you breathe in goes from, so here's the alveolus here, and here's the capillary. That oxygen is going to go from here into the bloodstream, and when it leaves that, the concentration of oxygen is going to be the same here as it was here. And so it's going to diffuse until it reaches equilibrium, and it's actually very quick. Carbon dioxide is actually going to go in the opposite direction. So carbon dioxide your body was producing is taken up to the lungs, where we're going to exchange it for oxygen. So CO2 goes from high to low, so it's going to go from the bloodstream into the alveolus, and you're going to breathe it out. Oxygen's going to go in the opposite direction. Well, what type of things could affect the rate of simple diffusion? So I'm going to show you some examples of, and I apologize for the glare, but sometimes it's hard to get pictures. So I took a plate that had agar in it, and I created some wells, and I filled the wells with two different dyes. And so this one here, this is methylene blue. This one here is called potassium permanganate. What you'll notice is I allowed this to diffuse for a certain period of time, And what you'll notice is this one did not diffuse as great as this one. So what do you notice? There's a difference in their molecular weights. So substances that have lower molecular weights will diffuse at a faster rate. So that's one thing that can influence the rate of simple diffusion is the size, molecular weight. The lower the size, the faster it diffuses. So that's one thing. Well, I also did a similar thing, but then I compared putting plates, setting them at room temperature versus in a 37 degrees Celsius incubator. And what you'll notice is that at 37 degrees Celsius, these are a little bit bigger, is because they diffused faster. So the warmer the temperature, the greater the rate of diffusion. So that's another thing that influences it. Third thing would be concentration gradient, because as we defined it, is we're moving substances from a high concentration to low until it reaches equilibrium. The greater the concentration gradient, the faster the rate of diffusion. So let's make a list. Okay, so we have it. So what are some factors that influence the rate of simple diffusion? Concentration gradient is one. So the greater the concentration gradient, the faster the rate of diffusion. So write that down. Number two, size of the molecules. So the lower the molecular weight, the faster the diffusion rate. Or we can say the higher the molecular weight, the slower the diffusion rate. And then the last one would be temperature. So the higher the temperature, because that actually increases that rate of random molecular motion. Things start moving around a little bit faster. So the higher the temp, the greater the rate of diffusion. The lower the temp, the lower the rate of diffusion. So those are things that can influence the rate of simple diffusion. Now, what about facilitated diffusion? They both have diffusion as part of their name, so there's got to be some similarities there. Well, the similarity is that they both involve substances going down its concentration gradient. It's going from high concentration to low until it reaches equilibrium. The difference, though, is with facilitated diffusion, the diffusion across the membrane is with the help of a carrier protein. It needs some sort of molecule that's embedded in the membrane that permits it to move across that bilayer. So things that move by facilitated diffusion, those are polar substances. They're hydrophilic. They can't go directly across the membrane, so they're going to need some sort of carrier protein to get across. So for facilitated diffusion, again, it's going to go from a high concentration to low until it reaches equilibrium. A great example of something that moves, there's a lot of things that move by facilitated diffusion, would be, say, the movement of glucose across most human cells, which you see right here. Okay, so you see that. Now I want to show you a graph that shows you a difference between simple diffusion and facilitated diffusion. So on the x-axis, you see concentration. On the y-axis, you see the rate of uptake, or that's the rate of diffusion. As you increase the concentration, what you'll notice with simple diffusion, what I had said before, is you'll notice it's steadily increasing. So I'm just going to go over it in blue. So you see it goes up and it's linear. The greater the concentration gradient, the greater the rate of diffusion. However, if you look at facilitated diffusion, you see initially, yes, as the concentration gradient increases, the rate increases until a certain point where then it plateaus. So question is, why does it plateau? Well, we have a limiting factor here. It's the number of carrier molecules. So this point right here, the reason why we can't go any faster is because all of the carrier molecules are bound to that molecule that they're carrying. They can't go any faster. So the only way you'd be able to increase that rate even more is to add more carrier proteins. So things that will influence the rate of facilitated diffusion would be concentration gradient, would be one, and another would be the number of carrier proteins. Okay. Give you an example, a real life example of where, say, you just ate and your blood glucose levels go up, and we want that blood glucose levels to go back down. We want our tissue cells to take it up and use it for energy, store it away for later. What happens is the hormone insulin will actually increase the number of glucose transport proteins that we have in things like as adipose tissue and skeletal muscle. So adding more carrier proteins will allow us to increase the rate of diffusion and help to get that blood glucose levels back to where it's supposed to. So by changing the number of carrier proteins, that can influence that rate of simple diffusion. Plus you have the additional thing is you got a lot of glucose because you just ate. That would help to increase the rate of diffusion too. So we're going to stop here. The next recording is going to cover another passive transport process referred to as osmosis.